I'd just like to welcome our next speaker, who's Dr. Nicola Walker, who works for AB Vista. She's been with us for, must be a couple of years now.
3 years.
3 years now, time flies. And she basically heads up our ruminant R&D development work. So I'll let Nicola introduce herself. She'll be better at it than me. Thank you.
Okay, I'm a lot smaller than Rick, so I've got to get the microphones kind of adjusted here. Also, for those of you who can't possibly see me because I'm so small, I'm hidden by the podium. I am actually here. I'll wave occasionally. For those of you who are not familiar with AB Vista, I think the best way to describe them as that they're an enzyme company. They're a feed enzyme company. They're probably the third or fourth largest animal feed enzyme company in the world. And we actually work with our sister company, who's AB Enzymes, and we actually take their enzymes and put them into animal feed. So if we actually look, just a quick biology 101 refresher for those of you who might not know what an enzyme is. Essentially, they're proteins, so they're proteinaceous particles. They fold into a tertiary or quaternary structure, and how they fold dictates their active site or binding site, which the substrate will attach to, and then the enzyme will degrade that substrate. They're classed as biological catalysts. They have very specific reactions for the substrate that they work on. They can be affected by environmental factors, so they always have a specific optimum temperature or pH. They interact with cofactors and coenzymes that can affect their activity. And there can also be certain inhibitors that will reduce their activity. So that's kind of biology 101 finished. Now, enzymes are actually used every day. There's a wide range of different different industrial applications that are used. Huge range of different enzymes. They always end in the A's at the end, so that's when you know you're working with enzymes. We have starch-degrading enzymes, amylases, protein, proteases, cellulases that break down cellulose, xylonases, gluconases, the list goes on and on. I don't know how, oh, there's a gentleman there wearing a pair of Stonewashed jeans. They were actually stonewashed by enzymes. You maybe didn't know that, so just as a little aside. They also use them in the pulping industry so that we can actually break down the wood to make it into paper. We use it in beverage, so we can use it in apple pulp and so on to break down the pectins in the pulp. I use it in baking and so on and so forth. But where I'm really interested in is how we can actually use enzymes in animal feed. So this is, for those of you who know me, this is probably one of the few times that you'll actually see that two-legged feathered variety on a slide that I present. I don't normally speak about chickens. I like to eat chickens, but that's about as far as I go with the chickens. Pigs, slightly different, like pigs, like bacon, and I actually do some research on the pig side. So enzymes in monogastric systems, these have been used since the 1980s. Very well established, a large amount of supporting data to show that they work, they're beneficial. Their use is primarily driven to try and improve feed efficiency, to maximize what that animal can get out of the feed. Also used for removing anti-nutritive effects and to reduce environmental effects. So that's reducing phosphorus excretion or nitrogen excretion. The main types that we'll use are carbohydrates. So these that break down carbohydrates. So we have xylonases for the hemicellulose components, gluconases, mannanases, and amylases. Phytases being used predominantly in the poultry industry, and proteases too. So what are these benefits for the monogastric? If you actually look at the rumen themselves, the rumen bugs provide a large number of these enzymes. So if you looked in the rumen, a lot of the bugs there provide the cellulase activity, the xylonase activity to help break down the fiber portion. You'll also find a large amount of phytase activity. But for the pig or the chicken, because they're built the wrong way round, in my opinion, you find that they don't have those bugs. And the feed actually has to pass to the lower gut where the bugs are present to be able to remove any of these anti-nutritive effects. As a result, you get poor digestibility. This can lead to loss in performance, and you get increased excretion, particularly of phosphorus, which is a problem when we're all trying to be super environmentally friendly. If you actually look in the monogastric industry as well, these are generally not over-the-top feed additives. They don't put them on top of the feed. They'll actually formulate them into the ration to get least cost value. So quite different in how most ruminant people look at feed additives. If we look in terms of the most common used, we find that phytase is perhaps the best known. It's involved in the destruction of phytate. This is contained within the plant itself. We know we can reduce feed costs and lower phosphorus excretion. Silonases, beta-glucanases, very important in the breakdown of plant fibre for the monogastric. Without this added enzymes, they can't do this efficiently. And we also find that we reduce the viscosity of the digester, so that the passage rate is altered. We find that we can lower feed costs, get better feed efficiency, and also we have less problems with wet litter. So as a chicken farmer, that's always a big issue, because who wants to deal with wet litter? Starch degradation from amylase, we can use this to actually help improve starch breakdown, better feed efficiency, and proteases, again for the breakdown of protein or better feed efficiency. For some of the work that we're currently looking at, we're also looking at proteases to put in silage inoculants as well for improving starch digestibility. So if we then move on and look at the actual evolution of the feed enzymes market, well, the ruminant is kind of down here. We're very much at the R&D introductory stage. Aquaculture is even better advanced than ruminants in the use of enzymes. And where you have most of the growth or very mature market is very much into the poultry or swine segment. And again, very much swine falling behind poultry and carbohydrates below phytases. If we now look at feed enzymes for ruminants, this is actually a farm that's in Florida, where we actually got enzymes we're feeding to these cows. Well, if we actually look at the past history, when I was a very young lab rat, which was a very long time ago, in the 1990s, so you can work out my age if you want. Yes, I am that old. And we were looking at work of using enzymes to try and improve feed efficiency in ruminants, even back then. Most of the research was on xylonases and cellulases, and this was to maximize forage digestibility. But, you know, there was a large degree of variation that we were finding in a lot of the trials that were being run both in our lab, but also in other labs. And a lot of this came down to different enzyme compositions. Some people were just putting cellulases in, some people were just putting xylonases in, some had different ratios. There were different dose rates, and we also had some dietary interactions which seemed to affect the way in which the enzyme would work. But one of the biggest prohibitive factors was cost. At that time, if we were going to feed a dairy cow feed enzymes per day, it would cost about 35 pence per cow. Now, I don't know any farmer that would pay 35 pence for any feed additive. I'd like to find them, because then I could sell them lots of stuff at very expensive prices. However, things have changed. Because these enzymes are being used in a wide range of different industrial applications, there's been a lot of work and research gone in trying to improve the yield. So the costs have significantly decreased. Milk and feed prices have increased. So as a result, we really want to maximize what we can get out of what we're feeding. So we really do have that need or drive to improve feed efficiency, and particularly to improve forage utilization. And one of the other things that's been key, I mean, this is 20 years' work, we actually have a better understanding of what's going on. We have a better understanding of how to feed, how to apply. We also have a better understanding of which diets may work best. So we look, and one of the biggest things, I'm a rumen microbiologist, don't ever confuse me with a nutritionist, because I'm not one. And where I'm really interested in is what the bugs can do to improve fibre digestion. If you actually look, the anaerobic fungi play a very important part. They're the initial colonizers of the fiber. So their role is to actually attach to the fiber and help pull it apart so that the bacteria can get in. So they have a role to play in speeding up rumen fermentation and fiber digestion. They have both cytolytic and hemicytolytic activities. They make up 8% of the rumen microbial population. And a lot of people tend to underestimate their actual role or function and what they do. They're also affected by pH. So in diets, for instance, where you're feeding a lot more concentrate, you'll generally not find a large number of fungi present. If we actually look at the protozoa, these account for up to 33% of fiber degradation in the rumen. So they are really important. Most people tend to think of them as just recycling bacteria and being involved in recycling nitrogen. But there's actually a large proportion of the protozoa that are involved in fiber degradation. If you actually defaunate, and there's some very interesting techniques that involve Vax Hoover and some formaldehyde to defaunate fistulated sheep, but I'm not going to go into it. But from those studies, we've seen that if you take all the protozoa out, fiber digestion is significantly reduced. So they really have a significant role to play. They, again, just like the fungi, are affected by any drops in pH. So, you know, we're seeing a pattern here. All the bugs that are involved in fiber digestion negatively affected by any drops in pH. Then we have the bacteria. As you can see, they're very interesting in terms of there's only 3 main cytolytic bacteria in the rumen: Fibrobacter succinogenes, Rhinococcus albus, and Rhinococcus fabaceans. So that's only 3 different species that are involved in fiber digestion. There's a whole range of hemicellulitic bacteria, such as the Prevotella and Butavibrio. And again, fiber digesters significantly negatively affected by any drop in pH. So how can these actually work with the enzyme? If you look here, we have a schematic showing the Rumnococci and the Fibrobacter attaching to the plant cell wall. We have the Butavibrio here that are involved in hemicellulose degradation and the Bacteroides and Prevotella. And what actually happens is when we actually put these in with, or we've supplemented the animal with exogenous enzymes, is we find that we can increase the colonization and numbers of these bacteria here that are involved in attachment to the plant wall, also the hemicellulose. So there's a degree of synergy that we get from the enzyme and from the bacteria or the microorganisms that break down the fiber. If we actually look here, we have a picture of wheat straw. On the left, this is untreated wheat straw. On the right, we have enzyme-treated wheat straw. Hopefully you can see there are holes that have appeared in the surface of the fiber. And basically, these are pits. So these are the pits which open up in the surface of the fiber through the enzymatic action that allow the bacteria in. The analogy that I like to put with this is it's kind of, if you look at that particle of wheat straw, it's a bit like a building. What the enzymes do is they break the windows in the building that allow the thieves or robbers, the bacteria, to gain access. But you don't collapse the building in any way. You still retain that structural integrity. You just get all the good stuff out from the inside. Because if you end up destroying that structure, you lose physical effective fiber. We also find, because we've actually made these pits, we increase colonization. On the right is a control sample of maize silage colonized by some Fibrobacter. And this is with in the presence of the enzyme. And you can see far more bacteria actually attached. So what we're doing is we're speeding up that colonization process. This is some work that's been done for us by BioParametrics. They have a gas production in vitro system from which they can model rumen fermentation. And what we've actually seen that they fit the gas production profile to their model. From that, they can determine what the effect has been on the predicted ME. They can work out the lag time to digestion, and they can work out the effect in terms of fermentable NDF. Well, actually, this is some whole wheat crop with control. Our predicted ME has gone from 9.6, and with the enzyme, it's gone up to 10.5. Grass from 9.8 to 10.4, 11.7 up to 12.7 for the maize silage. Lag time in every instance has been decreased. And as a result, if we look at the percentage of fermentable NDF, we're increasing this by anything between 4 to 5%. So this is what the enzyme can do. We've also looked, this is a study that was done in some lactating animals to look at the effects on the rumen flora. And we find that if we look Fibrobacter succinogenies, Ruminococcus albus, Ruminococcus flavifaciens, their numbers in dark green for the enzyme supplemented animals are far higher, significantly higher than in the control animals. We're also seeing a numerical improvement in Prevotella, a hemicellulose digester. What was also interesting is Ruminococcus amylophilus, which is not a fibrolytic organism, but is one of the first colonizers of the grain. So what we're doing is we're making holes in the surface of the grain that is allowing that bacteria in. We also found an interesting interaction between fibrolytics and selenomonids, but that's a completely different story. So if we actually look in terms of performance, okay, I've kind of tried to show you the mode of action, what the enzyme does, how it interacts with the rumen flora, how we can improve fibre digestion and utilization. This is a study that was done at the University of Reading a good while ago, when I was still a young lab rat, and we were working on a consortium with them. We have control cows. We have cows in which the TMR had been treated with enzyme. We have cows in which the concentrate portion was treated with enzyme, and we have enzyme that was infused directly into the rumen. And what we can see is that we're getting a significant improvement in total tract dry matter digestibility when applied to the TMR. We have more duodenal flow of nitrogen due to an improvement in microbial growth. We see a huge improvement in terms of post-rumen NDF digestibility, and we also see a reduction in fecal starch. Retention time of corn stover was decreased, and we got about 1.5 litres more of milk. And this was a Latin square, so it was a 4-week study with cows being swapped over between the different treatments. This is another study we ran in Canada with Karen Boschman's lab. We had 60 early lactation Holstein cows split into a low and high dose of enzyme. Interestingly enough, in this study, we didn't get the milk yield. They stayed the same. But a lot of this came down to the diet having been over-formulated. It was actually formulated for 39 kilos per day. But what we did see was a significant decrease in dry matter intake. And as a result, feed efficiency was significantly improved. And it was a dose-dependent effect. And this is one of the things that you have to remember when you're actually working with enzymes. It's very much dose-related. Too much is a quadratic response. You can actually go over, and you can have a negative effect on fibre digestion. And one of the reasons that is because those pits or holes that allowed the bacteria to get in are too many, too numerous, and it basically makes the surface of the fibre like Teflon, so anti-stick. So you have to be careful on the dose rate you use with these type of additives. So in that study, we saw that reduction in dry matter intake. There was no impact on milk yield. We improved feed efficiency at the lowest dose by 5%, and this increased to 11% at the higher dose. We improved the percentage of protein, improved weight gain as well, so there was an indication of less mobilization of the animal's reserves during early lactation, and there was a potential reproduction effect as a consequence. We actually have one vet in Utah. He was using the enzyme on dry cow rations, so that he is actually setting up the rumen before early lactation. He's finding he's getting less issues with DAs, less problems with ketosis, basically because these cows are basically in the mindset to eat. Their intakes are higher. They tend to, the rumen's primed basically for fiber digestion when they actually hit early lactation diets. This is a meta-analysis, so being pointy-headed people, we always like to do meta-analyses, conducted by Yoon, who did a lot of the early work on enzymes in Canada. This was using 10 published studies, using xylanase and cellulase mixtures, and then running a meta-analysis on all the information to see what the results were. So in terms of enzyme treatment or supplementation, we find we get a significant improvement in dry matter intake of about ½ kilo. Milk yield was significantly increased by 2.3 litres. Energy-corrected milk yield was also significantly improved. There's no effect on butterfat, no effect on protein. Milk fat numerically went up. Milk protein was significantly increased. But where the big effect was on feed efficiency. So we're always improving, significantly improving feed efficiency in terms of milk yield over dry matter intake, and numerical improvement on energy-corrected milk. We've actually just finished running a study in the US where we got a stupid increase in energy-corrected milk, such that, we're going back to recalculate because I don't believe it. So what exactly do the enzymes do? If we actually add the enzyme to the feed, this causes the pits to form on the surface of the fiber. We then, the animal eats it, we increase colonization by the bugs, we increase the number of the cellulitic and hemicellulitic bacteria, we decrease that lag time to digestion. The cow gets more out of what it's eating, we get better feed utilization and efficiency, and we're actually increasing what was the predicted ME of the feed. So where we're actually having, and as a consequence, we're getting increased milk yield and feed efficiency. On the commercial studies that we've run in the US, what we're finding where this works really well is down in Florida, where there's highly lignified forages. And we're also finding, even in the Midwest, where they're feeding BMR, corn silage, that you would think be readily digested, we're still improving and speeding up that digestion process. So providing we manage to get the balance of the ration right, it does seem to work on a variety of different forages. And for me, I think what we can see is that the future of enzymes and dairy cow nutrition are hopefully very bright, if I look into my crystal ball anyway. Thank you.
Hi. When's the enzyme added to the silage? Is it in siling or is it just before feeding?
No, and sorry, I'll stand back here. You'll actually find the use of enzymes is two different applications. One is in the silage inoculant, where the enzyme is actually releasing sugars or opening up the fiber for the bacterium that are used in the inoculant to help and speed up that ensiling process. What we're doing in this instance is we're actually treating the feed in the mixer wagon, and then we're feeding it out. The dose rates are completely different. What you would use, the levels that you would use as a silage inoculant are far less, because you've got a lot more time for it to work or to have an effect than it would be when you're actually putting it in feed that you're feeding out to the animal.
Okay, thank you very much, Nicola, for a very interesting talk. I think everyone will agree that the use of enzymes in ruminants is a relatively new thing, and I think it's only the beginning, and there's a lot of work to be done in this area, and will be done over the next few years. So that really brings us to a close of the pre-seminar session, and again I'd just like to thank everyone for attending and coming early to register for the large herd and attend the pre-seminar.